start with Dr. So we will start with Dr. Munia Ganguli. She will be talking about non-viral delivery of nucleic acids to the muscles. Dr. Ganguli, please go ahead.
Yeah, am I audible and are the slides visible?
Yes, you are audible and the slides are visible as
well. Okay. Good afternoon, everyone. And
so sorry to interrupt, but could you put it in slideshow mode?
I did that. Ah, okay.
Now, thank you.
Yeah, now it's fine. Yeah, okay. Yeah. So good afternoon, everybody, and I'm really, happy to be here today and very honored to be able to talk about, some of our research. And thank you, Professor Bhattacharya, for giving this opportunity. And, what I'll be talking about today is about non-viral delivery of nucleic acids. Primarily DNA is what I'll be talking about, but the same applies in some way to RNA as well. To the muscle and in the context of GNA myopathy. So I'll spend first few slides explaining the process of non-viral gene delivery or non-viral DNA delivery. And I'll be using these terms a little interchangeably. And this is mainly because, today's audience is also, comprising many non-scientists. So I'll give a little bit of introduction on one second. Yeah. On the whole process, and then I'll be moving on to some of the data. You do gene delivery in order to achieve what is called gene therapy. So what is gene therapy? It is nothing but transferring a right gene instead of a defective gene that the cell already has. You transfer a right gene to produce a right protein. And while transferring the right gene is one way of doing gene therapy, it could also mean that you have to block a gene that is not functioning properly. That also constitutes gene therapy. And in the near past, there has been a lot of work on what is called gene editing. So that's also a form of gene therapy where you are cutting out portions of the wrongly functioning gene and correcting it. So all of this constitutes gene therapy. And how does one do gene therapy? For doing gene therapy, you cannot introduce the DNA or RNA for that matter, any nucleic acid. You cannot introduce from outside in sufficient amounts if you do not have a delivery system. The nucleic acid or the gene does not enter without a delivery system, does not enter efficiently without a delivery system. So when one talks about delivery systems, there are virus-mediated delivery systems. So the viral systems is what's shown here, and also non-viral systems. So non-viral is what I'll be talking about today. You can directly inject the delivery system into the body, which constitutes the in vivo gene therapy. You can take out cells isolated from a patient, modify them in vitro, and then put them back into the body, which constitutes the ex vivo form of gene therapy. Why non-viral gene therapy? If you look at these charts, if you look at the chart at the left side, the 2019 one, you can see that the different types of vectors or the different type of delivery agents that are used for gene therapy are primarily, constituting viruses. So different types of viral vectors, and I'm sure Arkasubhra is going to, give an overview of that in the next talk. But with time, and especially, when the COVID came and new vaccines, the mRNA-based vaccines came about, there was a lot of interest in using non-viral methods of transferring these nucleic acids. So by 2021, you can see that other vectors, the not known vectors, the RNA transfer, these are actually, different kinds of non-viral methods. And the non-viral methods have certain advantages. They are less toxic, they are easy to prepare. A large size of nucleic acid can be introduced. There isn't theoretically not much of a limit in terms of how much nucleic acid or DNA you can introduce. You can can target them, the surface you can modify and target them to the cell type that you want, and they are also less immunogenic. So now let's look at what are the steps of doing this non-viral gene therapy. The first and most critical step is to package the DNA using different kinds of chemicals. So these chemicals constitute the non-viral method. And I will call this this packaging, the material that comes after packaging, as a nanocomplex. Okay, so the DNA or the nucleic acid and the chemical, they are mixed in a certain way to form very small sized particles called nanocomplex. These are the ones that you inject in the body. They will then enter the bloodstream, they will reach your target cell and then go to the nucleus and express the gene. That's the idea. But in doing so, there are a large number of barriers that the nanocomplex will face. First of all, it goes to the liver. When you inject it, it goes to the large amounts go to the liver and they're sequestered there. If the size is very small, they can be just cleared out by the kidneys. When it is traveling through the bloodstream, when you inject, it travels through the bloodstream, and the proteins in the blood can form a layer on the nanocomplex and alter its properties. So these are the organ level barriers. Then next, like, it has to come out of the bloodstream, it has to go transport into the target cells. So there are sub-organ level barriers. And then it has to go inside the cell. It has to be taken up in large amounts inside the cell. And inside the cell also, there are several barriers, like coming out of the endosome, targeting the nucleus, and so on, that it has to undergo. So in order to overcome these barriers so that large amounts, sufficient amounts can be effectively transported to the cells, the carrier design, or the gene delivery agent design, the nanocomplex design, is very, very important. The size, the shape, the surface modification, these are very critical factors, and you have to intelligently design. You have to do a bit of trial and error to get the right system that will effectively overcome these barriers. So that is what the struggle is all about in the non-viral gene delivery space. So what are these materials that one uses as non-viral carriers? They could be lipids, they could be polymers, they could be inorganic particles, biological vesicles. Some of the pictures are given here. They, by and large, they look different, but the basic principle is the same. It could be a combination of these different carriers also, but the basic principle is the same, that they interact with the nucleic acid, form very small sized particles. So that's about the introduction. Now, what are we doing in this space in the context of GNE myopathy? So our aim here is to design a non-viral gene delivery system that will go to the muscle. Okay, so to do that, there are three components that I'll be talking about here. Primarily two components I'll cover, but I'll mention the third one as well. The first component is the nucleic acid itself. So that is the a plasmid that has the GNE gene. That is my gene of interest. But before we actually send the GNE containing plasmid into the cells, we can also test our gene delivery system by using what is called a reporter gene. So all the data, little bit of data that I'm going to show, or the work plan that I'm going to show, initially we will be using a reporter gene as a mimic for what we are going to eventually deliver. And why so? Because it's easy to assay. So there will be a fluorescent, so usually one uses what is called a green fluorescent protein, and that fluoresces, so you can easily assess whether your gene delivery agent has worked or not. Okay. The second component is the system itself. The system itself, there are two systems that I'll be briefly talking about. One is a peptide-based system that we have developed in our lab for other purposes, but should work the same way here, and a polymer-based system that we are newly starting with. The third component is a muscle targeting moiety. We are going to use a peptide, and that is to take the system directly to the muscle. So now we are going to make combinations of these. So we are going to attach number 2 and number 3 and mix it with the DNA to form the structure that you see on the left. That's the agent with the targeting moiety that is attached on the surface. we can also first make the nanocomplex with the, with 1 and 2, and then coat the muscle targeting peptide on the surface. The idea is that you make a nanocomplex, you decorate the surface with a muscle targeting peptide, and you do that in different combinations. So let me now go through these two systems, the in number 2 here, that what I mentioned, a peptide-based system and a polymer-based system. So this is the peptide-based system that we have developed in our lab. It's an amphipathic peptide. It has some positively charged residues, positively charged arginines, and some hydrophobic uncharged residues. And we have found that this peptide, it can form nanocomplexes with different kinds of biological molecules, not only DNA or with siRNA, with some therapeutic peptides, with some polysaccharides. It can, through electrostatic interaction, charge-charge interaction, it can form these nanocomplexes, and they can be taken up by the cell very efficiently. It's also easy to synthesize, it's non-toxic, and large-sized cargos can also be taken up. And this is a small picture of NGP9 peptide, going into skin. So this is our first system. The second system is a polymer, and Professor Mishra had mentioned about this. This is a class of polymers called polybeta amino esters. These also have a cationic charge, and they are biodegradable. And the structure is such that many kinds of designs are possible. By changing the monomer in the polymer, by changing the units at the end, one can design in different kinds of polymers. They are they have low toxicity, and they are also highly efficient as compared to well-known polymers like polyl lysine, PLL, or polyethylenimine, PEI. The PBAE polymers are far more versatile. They are they have low toxicity, and they're very efficient in delivering DNA. So this is going to be my second system. Now, coming to the point number 3, that is the muscle targeting peptide. Okay, so we have looked through the literature and searched for a number of different kinds of peptides that will exclusively go and bind to the muscle cells and help in transporting the cargo. So we have chosen few of them. One is a skeletal muscle targeting peptide that has been coupled with dendrimers for gene delivery. Second one is a alpha dystroglycan binding peptide. So that's there on the muscle surface, so it can go and bind on the muscle cells. And the third one is derived, it's a very recent work. It's derived from AAV capsid variants, and it's an integrin binding peptide that also has been shown to have muscle directed gene delivery properties. So this is my third system, the muscle targeting peptide. So coming back to the schematic that I had shown, the nucleic acid, yeah, we'll start with the reporter, but eventually we will be delivering the GNA gene as well. The second one is my, the non-viral gene delivery system, the MGP9 peptide or the PBA polymer. And the third one is the muscle targeting peptide. And we are going to choose one of these 3 and then make these combinations and create these nanocomplexes that will deliver the gene. So where are we right now? We already have point number 2. The system is ready. Point number 3, we already have the designed peptides. We already have gotten them synthesized. We are yet to start experiments with those. But once we do that, we are going to characterize the nanocomplexes, find out what their size, charge, surface is going to be like, and then put them in the cells and check for their efficiency of gene delivery, their cellular viability check, and so on, and also put them in mouse model to see where these nanocomplexes are distributed. If you do an IV injection, where do these nanocomplexes go? So once we are done with that, and all of this using the reporter gene, so that you can easily assess whether your system is working or not. Once that happens, in parallel, we'll be also having the GNA construct, and then we will be sending it, we'll be doing those experiments with the GNA construct and see if the GNA expression is there or not, and a set of experiments we are going to do to assess that. Just a little bit of preliminary data. So as I said, that we still haven't started experiments with the muscle targeting peptide, but with the basic nanocomplex system, this is with the peptide that I mentioned, and this is in mouse muscle cells. We do see that, the green that you see in the pictures, these are the reporter gene that I'm talking about. So we are able to standardize the condition for the expression. For the polymer system, we had to do, for the peptide one, we had already been working on for quite some time, so we kind of knew what conditions to use. For the polymer system, we had to do some, trial and error to get to the right size, right charge, and so on. And this is the data for the preliminary data for how this polymer system is transfecting, or how it's carrying the nucleic acid, the reporter nucleic acid, into the mouse cells. And the lower bottom panel is with the polymer, and you can see that there is a lot more expression as compared to what lipofectamine, which is a gold standard for sending nucleic acids to cells. And we are very excited with this, and we are now going to modify these systems with the muscle targeting peptides, and we have to then see whether we can exclusively make it, or at least majorly make it, go to only muscle cells and not to other cells, first in vitro. And if that works, then we will be moving on to in vivo delivery. Yeah, so I think I'll stop here, and I would really like to thank Professor Alok Bhattacharya and Professor Sudha Bhattacharya for giving me this opportunity, WWGM and the community, for all that they're doing, and ICMR for funding. The funding has just come in, as Professor Bhattacharya mentioned. From February onwards, we are working on this, and my students Divya, Franklin, Betsy, Anupama, and Rajpal for taking care of different aspects of this work. Thank you very much, and I'll be happy to take any question.
Thank you very much, Dr. Ganguli. We can take one or two questions. Others can, put the questions in the chat box, and probably you can answer them gradually. Any questions?
Hi,
Yeah,
I'm Shri
can I?
Darshini. Oh,
Okay,
sorry.
can I ask one thing? Surajit
Yeah,
here from Cultivation of Science. Actually, I am
please go
working
ahead.
on the delivery vehicle, and as per my discussion and experience with Professor Maigate, Matthew Wood from Oxford University, and Vice President of Alnylam Pharmaceuticals, Boston, and the pioneers, those who are working in this area, they strictly told that CPP peptide-based delivery vehicle, moreover having too many guanidinium group, better do not use, because ultimately did not work and giving too much toxicity. So they all are living to work in this area. And recently we visited Sweden in a symposium. Some of the pharmaceuticals company told that we will not be interested to fund your project if you work in this area.
Yeah, so the issue with the cationic peptides is, if you have very large stretches of arginine, we have tweaked the structures in such a way that we are also interspersing it with hydrophobic residues, making it secondary amphipathic, so that one phase has arginines, one phase has hydrophobic moieties. So it's a little different from the 16-mer, 20-mer pure polyarginine systems that have been used for a lot of experiments. So that's a little bit of a difference. We also have a polymer system too, which is showing, in fact, the initial studies show it's better than the peptide system. So we'll be working on both of them. Yeah.
But that polymer also in vivo system, you have a ester bond. How will you protect from the esterase?
Yeah. So, but that's the balance that you have to strike. If you do not have that, you will not have a biodegradable polymer, right? So you have to make it biodegradable to reduce the toxicity, but you also have to stabilize, so that the system eventually reaches the target organ. Yeah.
Okay,
Great.
thank you.
Thank you. We can take one more question. The hand has been raised by Shri Dharne. Shri Darshan.
Shri Darshini.
Shri Darshane. Please go ahead.
Hi, I'm Shri Darshini. I work on Alzheimer, and The problem with the progressive neurodegenerative disease is that the disease is already, is diagnosed in the late stage. When the disease progressed. And the problem with the gene therapy is that all the other pathology take over, and the gene therapy, at that point, gene therapy doesn't work anymore, because amyloid aggregation, even in the GNE myopathy, which is one of the hallmark. So is the gene therapy for GNE myopathy will target only the preventative patient, or is it possible to, for the treatment or therapeutic cohort, people?
Yeah, so I guess I'm probably not the best person to answer this, but my idea would be that it has to be at an early stage, yeah, so that other pathologies do not sort of take over. But maybe Professor Bhattacharya can add something to that.
Dr. Alok Bhattacharya, would you want to add something?
Yeah, it's very, this is something that goes on all that. And quite often the clinicians who are designing a clinical trial will like and patients to be fairly in the early stage of the disease, so that, they can actually see. On the other hand, in some recent studies that I was looking at, people actually tried fairly late stage of the disease. Because sometimes in a late stage, small changes can be actually measured, and that can be very, very useful. And the patients at the late stage do need such a thing. But there is also, I think, everybody thinking of a long-term basis combination therapy, where you do, let's say, small molecule gene therapy, etc. You also probably give us a cell replacement kind of a system. So that the damaged cells which cannot be repaired with are also replaced. The answer is both happens in both directions.
Thank you so much.
Great. Thank you. Any further questions you can,
yeah,
type it down?
There are some questions on the chat box, and I'll reply to that.
Perfect, Dr. Ganguli. Great. So we'll head on to the next speaker, Dr. Arkasubhra Ghosh. He will be talking about gene therapy for muscle diseases, and he'll be giving an update on the current status of what's going on. Dr. Arkasubhra Ghosh, please go ahead.
Thank you. So I hope my slides can be seen.
Yes, the slides can be seen.
Great. So thank you, Alokta and Shilpi and WWGM for the opportunity to speak today. So I'll give a quick update on what we've been up to on our work on muscle diseases, and particularly in GNE myopathy. So this is a slide I always like to start off with, is the gene therapy roadmap for the country. So essentially, we need to focus on all of these areas that I've highlighted, understand the regulatory processes and ensure ethical practice, develop the vector production facilities so that you can make sufficient amount of the drug, which is of clinical grade, and which can be tested for toxicity in preclinical models and then go on to human trials. The third, of course, the most important thing is to identify the clinical teams and the appropriate infrastructure, because gene therapy trials are usually very long and they have to have a long-term follow-up. Then what would be useful for all the scientists who are working in these areas is to have integrated patient registries of disease phenotyping and mutations, so the genotype phenotype correlations, so that people can focus on function as well, particularly when you are trying to create synthetic transgenes which may have equal or better function than even the wild type. And most important, of course, is to create awareness within the government and other bodies to establish long-term secure funding for gene therapy research, for genetic testing of patients, etc. So the gene therapy landscape, I've just briefly mentioned some of the things that are happening here. My apologies to those who are not mentioned here. But in general, for muscle disease, eye disease, we work on it. There's a number of other institutes as well as industry folks who are currently working on it. Of course, a lot of interest in CAR T cell therapy, lots of new companies and lots of work in within the private institutes as well as government institutes. And in gene editing technologies, IGIB and others have been focusing on that. So the within this ecosystem, which is rapidly growing, of course, the major lacunae that remain are production capabilities of clinical grade vectors, availability of raw materials within India. For, if you ask any of the researchers who are here or whom you interact with, most of our reagents come from outside of India, they're produced outside of India. So if you can build that ecosystem of availability of materials in India, availability of resources and technical processes, that would really help translate some of these interesting in-lab results to clinical trials. So Dr. Ganguli has already explained what is gene therapy. So usually these are the 5 types of gene therapies. The one that we are going to talk about, which we work on in general, is where a normal gene or a healthy copy of the gene is put back into the cells and tissues of the patient or of the model, and we call it gene augmentation. So I'll be using AAV gene therapy, and I'll explain more of what is AAV. And this is a cartoon to show that we remove all the viral genes of the native virus and create a recombinant vector which contains only our gene of interest, which is a therapeutic gene. And the hope is that when you inject it into the patient, that therapeutic gene will produce the correct protein that will fix the disease. Now, the key components of such a gene therapy product are listed here. So you have 4 arms of any AAV vector. The first is the therapeutic transgene, the second is the capsid serotype, the third is the vector production process, and fourth is the clinical trial design. So you have to have all 4 of these developed for you to actually go into clinical trial and find success. So in case of the therapeutic transgene, there's the design of the transgene sequence itself. Then there is the design of the regulatory elements that control expression from the transgenes. There may be introns, etc. Within the capsid serotypes, now there's a lot of work across the world on finding various serotypes, finding mutant serotypes, which have a variety of tissue tropisms so that they can go and deliver it specifically to a certain tissue and also regulate the immune response to the capsid. So some of the work has focused on particularly reducing any immune response to AAV. The today is the vector production capacity. There's already the around the world, the demand for clinical grade vectors outmatches the ability or the production facilities around the world. So a lot of process and a lot of ideas and a lot of literally hard work goes into this vector production process, particularly producing vectors at large enough quantities that are needed for muscle gene therapy or hematological gene therapy. So we work with eye gene therapy, and that does not require as much vector to be produced at a time, 1,000 times more vectors than what you would require for the eye. So therefore, you have to think very differently when it comes to vector production processes for clinical trials. And finally, the clinical trial design itself. I think some of the speakers ahead of me have already discussed about it, and some to this is how do you design so that the trial gives you the most amount of information and is not bogged down or doesn't fail because of poor design choices. So patient selection criteria, the route of administration, the safety, efficacy, and all of these methodological things have to be designed with utmost care, and patient evaluation and follow-up process to evaluate efficacy. So what we work on is this AAV vector. It's very efficient in delivery. It's the gold standard for gene therapy being done worldwide. There are no viral genes in the recombinant AAV vector, and in vivo administration has usually been very safe, although at very high doses, which are currently being used, particularly for muscle gene therapy. There is a certain amount of toxicity, and we know about the patients that unfortunately had a bad reaction and passed away. So these are typically hepatobiliary responses because of overdose of the vectors. And finally, the vectors are fairly stable, but the prime reason why AAV is so safe is because AAV does not integrate into the host genome, so you do not have insertional mutagenesis. And the second thing is it's very tiny, right? So AAV, the wild type AAV, does not cause any known human diseases, although we have seen some associations in the past, but by itself, it does not cause any disease. It's a depender virus, and it simply sticks around in the cell for a while. Now, given that, we have been working on developing the AAV gene therapy for GNE myopathy, and this is work being done in the lab by my student Shubhadeep Sarkar. And what Shubhadeep did was he developed the GNE myopathy, the GNE AAV vectors, and then we tested them. This is under a CMV promoter, and you can see robust expression from the vector in a mouse muscle line C2C12. This is the expression fold change. We went on to do a preliminary study just in wild type animals. So we do not have access to GNE knockout animals. So we just simply to test out expression and safety, we injected wild type animals with our vectors. And we see robust expression of the GNE. This the top panel shows GNE expression in mouse muscle. And the bottom panel it's still GNE staining. It's not GFP, it's still GNE in staining. And you can see this fair increase in expression. The same thing is seen in the Western blots in muscle tissues, the same muscle tissues tested by Western blots. So this is using histochemistry, so the same results. We of course need to look at these vectors in a proper animal model, which replicates the disease. However, to develop the vectors further, we moved the GNE gene into a muscle-specific promoter, which is the SPC512 promoter. And we did the same thing. We tested those SPC512 promoter in the muscle-specific cell line, the C2C12 cell line, and we do see similar levels of expression of GNE. So this, the vectors are ready, they package well, they express. So if we are to go towards GNE gene therapy, what are the things that we have in hand, and what is it that we have to move forward towards? So definitely the muscle-specific GNE expression cassettes are ready, they work. The a number of recent papers have described AAV capsids, which are much better for transduction of muscle. We call them the MyoAVs. So a few of such vectors could be tried out for better expression in the muscle. And the third thing, of course, is large-scale vector production required for making clinical grade vectors. So right now at our laboratory in NNF, we have the bioreactor and chromatography-based technologies, which are already operational. The work currently is focused on improving the yield for from each of these processes so that we can eventually reduce the costs of production of the vectors. But for a trial, what we need to do is we need to prepare clinical grade batches and of these AAV vectors and use them for GLP tests and preclinical tests in animal models, relevant animal models. We need to initiate neutralizing antibody testing in the relevant patient group for evaluating potential trial participants. Of course, we need to have appropriate biochemical assays to assess gene expression and gene function. And then finally, we need to develop in detail all the clinical vector administration protocols, which should be in line with the National Gene Therapy Guidelines. So for the, like I pointed out, for the vector production, we have the clean rooms now, and these are photographs of the clean room and the operations. Currently, we are preparing to write our first clinical trial protocols. This is a video of the bioreactors that are used in the lab. This is not a video from inside the clean room, but a method development video that was done outside the clean room. It's so these are nanobioreactors. Of course, for something like GNE myopathy, probably the yields from such nanobioreactors is sufficient. But for other diseases such as LGMD or DMD, it'll be difficult to reach the requisite vector amount from one small bioreactor. Now, essentially, the bioreactor, this is a fixed bed bioreactor in which we see the cells and we do the transient transfection inside the bioreactor, and the vectors are produced. Some of it comes out in the media supernates, some of it is retained within the cell lysate, both of which are processed post bioreactor growth by chromatographic methods. So this is just to show the quality control, so that these are images of the transmission electron microscopy images of the AAV vectors produced. The white colored ones are the ones that have the genome inside, and the ones with the dark center are the ones that do not have a genome inside. So you can see that in our production, we find fairly high proportion of filled capsids, and there's fairly low background contamination. You can see this is the silver staining that shows the load and how all the background proteins have been removed. So with that, we have to think about how we are going to propose a clinical trial. This is a roadmap for how the clinical trial is to be conducted. If we are going for a clinical trial, these are all the regulatory approvals that we need from the Institutional Biosafety Committee, from RCGM, from the Institutional Ethics Committee, from the Gene Therapy Advisory and Evaluation Committee, and finally from CDESCO, which is the final authority to grant, clinical trial approval. All right, so we talked a little bit about it. So inclusion, exclusion criteria, whether or not we'll have a randomized control trial or an open trial. There are important considerations like establishing the neutralizing antibody titers to the vectors, because that is a cutoff that's very critical for patients. And of course, they should not have other ongoing trials or other surgeries, etc. So with that, I've come to the Yes, Rishabh, I'm almost done. So
Great, wonderful.
this this slide shows the status of the muscle gene therapy studies that we are currently doing. So there's DMD gene therapy, GNE gene therapy, there's LGMD. And for the first two, we have done the proof of concept studies in animals, and further grants have been written to take these studies forward. So right now, nothing is in clinical trial. We have some clinical trial designs already, but we cannot move further till we receive further funding. Same thing with a breakup. And with that, I'll thank all my sources of funding and my lab. Thank you so much.
Thank you very much, Dr. Arkasubhra, for this wonderful presentation. We have time for just one or two questions, and probably then others can type in their questions in the chat box, and later on you can answer to them gradually. Any questions?
Maybe I can ask a quick question, Arka. I mean, thank you for this talk. I mean, we are very impressed with all the roadblocks that are there, and we really have to work together to take care of it. Just quickly wanted to ask you, from all your experience with eye disease, you have some idea of the kind of AAV serotypes which will be safe to use in the Indian population, where the positivity in Indian population is low?
Yeah, that's an excellent question. So we are hoping to answer that. We currently have an ICMR funded study under which we are doing neutralizing antibody titering. So this is a study led by Dr. Swaminathan Setu in our group. He's an immunologist. And what he's doing is, so they've already standardized the NAV assay, and we are taking samples from eye disease patients, from muscle disease patients, blood disease patients, and trying to compare what is the prevalence of neutralizing antibody titers against 4 different serotypes, which include AAV2, 6, 8, and 9. So we will have a better idea. I can give you a much more informed idea of what the prevailing neutralizing antibody titers are in the Indian population in maybe a few months' time.
Thanks a lot.
I have a question. It's a nice talk, and the one of the major drawback in using AAV is the size of the
that's right.
Size of the gene
size
that
of the transgene. Yes.
But then GNE is around like 2 kb, I believe, and the promoter, and also it should be counted. I mean, including the ITRs.
Yes.
So, and also it affect the titer, which affect the infection. I mean, how I mean,
multiplicity of infection.
Yes. So what's the titer value? Because it will affect the efficiency of the gene therapy. What's the titer value have you reached? You have reached, and how will I don't know.
So yes. So essentially, you're asking in our production process, are we reaching the sufficient titers so that we can do GNE gene therapy? So yes, for GNE gene therapy, at least the initial studies will be local delivery to the distal muscles. So for that, we are already reaching the sufficient titers. So we are in the scale of 10 to the 13th vector genomes per production, between 10 to the 12th and 10 to the 13th per production cycle. So that is sufficient for a local gene therapy trial. Now, we are hoping that by this time next year, even with the bioreactors, will go up by another 5-fold at least. And we have written a lot of grants, etc., to the government agencies for a large-scale vector production facility. Dr. Alok is heading one such effort, and I have also written other efforts. So keeping our fingers crossed. So if those come through, then we will have the ability to produce vectors in the range of 10 to the 16 vector genomes per cycle.
Oh, thank you so much.
Thank you very much. So how should we proceed ahead, Dr. Alok, should we play the video?
Now let's play the video. I don't see him now.
Dr. Ghosh, there are some questions in the chat if you could address, please.
Certainly.
So Shilpi, I don't have the video, so
I'll play.
okay. Dr. Alok, please go ahead.
Yeah, I'm doing it.
This is Ram Kumar from ISER Tirupati. I'll be presenting the progress we have made in the last year in generating cellular models for GNE myopathy. This project is supported by WWGM. Also resources that we get from ISER Tirupati. So to begin with, there are two methods to derive skeletal muscle starting from pluripotent stem cells. So when we are able to derive skeletal muscle starting from patient-derived pluripotent stem cells, they will serve as preclinical models. The two methods are basically involving same steps, but different protocols that allow us to generate skeletal muscle in the lab, starting from stem cells. This is a published method, and this is a method which uses proprietary media components. And this is also
Dr. Alok, the voice is not audible. Dr. Alok? Dr. Alok, are you there? Shilpi, can you hear me?
Yes, I can hear you.
Okay, so the voice is somehow not audible.
Yeah, I don't know why Dr. Bhattacharya is not responding.
Alok is saying that to remove mute, then it will be audible.
It's not audible. It's just
Yeah.
simply not audible.
It is audible to Alok.
But not to you, no? It's not audible
No,
to you.
it's not
No, it's
audible.
not audible.
Yeah. Yeah, I don't know how to do it.
Maybe we can post the video later on if
yeah, we can skip it and then later on we can post the
because
video.
We'll upload it on YouTube. We'll do that. Yeah.
So
Yeah.
anyway, Trace, it's 4:50 and the session is
Yeah, that's right.
just
That's
towards
right.
the
So
edge.
we'll have to skip it. It's not audible actually. Yeah.
Okay, so thank you to all our wonderful speakers.